Okay, so I want you to imagine something. Picture a scientist. It's the mid-70s.
Right.
They know life runs on these tiny biological machines, proteins. They digest your food, they fight off
They
viruses.
do basically everything.
Exactly. And they know that what a protein does comes down to one thing, its shape. But here's the problem. They have no way of figuring out that shape.
Well, not without spending months, maybe even years, in a lab with x-rays, a huge bottleneck.
A total grind.
Yeah.
Fast forward 50 years, and that process, that PhD level effort, can now be predicted by an AI in minutes.
Not years, minutes.
Okay.
It's a staggering change. And it's the tech that just won the 2024 Nobel Prize in Chemistry.
So today on the deep dive, we're looking at Alphafold. We're going to break down how DeepMind cracked this 50-year-old puzzle, why the new Alphafold 3 is a whole different beast, and critically, why you still have to be careful. I'm the builder.
And I'm the conscience. Let's get into it.
So to really get why this is such a huge deal, we have to start with the basics. If I zoom way in on a protein, what am I actually looking at?
You're looking at a string of beads. At its core, that's all a protein is.
A string of beads.
Yep. In biology, we call those beads amino acids, and there are 20 different kinds. Your DNA is just the instruction manual for what order to put the beads in. So you start with this long floppy chain.
But the proteins in my body aren't floppy chains. They're, tools. They have a job.
Right, because that string doesn't stay straight. It immediately knots and twists and folds itself into a really specific, really complex 3D shape.
And the shape is everything.
The shape is everything. Fold it one way, it's an antibody. Fold it another, it's an enzyme that digests your lunch.
Okay, and that brings us to the 50-year-old problem, this thing called Levinthal's paradox.
It's basically a math problem. A single protein chain could, in theory, fold in billions and billions of different ways. I mean, an astronomical number.
More possibilities than atoms in the universe, kind of thing.
Exactly. If I had to try them all randomly, it would take forever. But in your body, it happens in milliseconds.
So for decades, computers just couldn't figure it out. And then Alphafold 2 came along. What did it do differently?
Well, DeepMind realized they didn't have to simulate all the physics from scratch. They could look for a pattern. The real genius, I think, was using evolution as a kind of a cheat sheet.
Ah, this is the multiple sequence alignment part.
Exactly. AlphaFold looks at the same protein across, say, humans and chimps and mice and fish.
Okay.
The genetic code changes a little bit over millions of years, but the protein's job, and therefore its shape, usually stays the same.
So if one bead in the string changes,
then another bead that's touching it in the 3D fold probably has to change too to compensate. It's called coevolution.
I see.
AlphaFold got incredibly good at spotting those connections. If two beads always change together across species, they're almost certainly stuck together in the final structure.
It's using history to solve a geometry problem.
You got it.
Yeah.
And it worked incredibly well. It essentially solved the problem for single protein chains.
But you said it's single protein chains. This sounds like a big caveat.
It was. Biology is a team sport, Proteins don't just float around by themselves. They interact with DNA, with RNA, with other proteins, and crucially, with drugs.
Which brings us to Alphafold 3, the new model. So this isn't just a software update.
Oh, no. They completely rebuilt the engine.
Ow.
The headline change is that they switched to a diffusion model.
Diffusion. Wait, that sounds really familiar. Isn't that the same tech behind those AI image generators?
It's the exact same underlying idea, yeah.
So you start with what, a fuzzy picture?
You start with a screen of random static, pure noise, and the AI has learned, step by step, how to remove that noise until a sharp image appears.
Okay.
Alphafold 3 does the same thing, but instead of pixels, it starts with a random cloud of atoms, and it denoises that cloud until it snaps into a precise, stable biological structure.
Wow. So it's not just following a chain anymore. It's sculpting a scene from a cloud of gas.
That's a great way to put it, yes.
Does that actually work in practice? I mean, for the really complicated stuff.
The examples in the Nature paper are pretty amazing. They showed it predicting how a protein physically grips a strand of DNA, not just two blobs touching, but the actual, molecular handshake.
And drugs. I assume this is a big deal for pharma.
It's a huge deal. Before, companies used software called docking to sort of guess how a small drug molecule might fit into a protein.
Wow.
It was rigid.
Like trying to fit a key into a solid metal lock.
A perfect analogy. Alphafold 3 treats the drug and the protein as one flexible system. It predicts the interaction, how the key actually turns in the lock. It's a massive leap from just predicting structure to predicting interactions.
It's a huge pivot, but DeepMind didn't just build this amazing model and keep it for themselves. They released the data.
And that's maybe just as important as the model itself, the Alphafold protein structure database. They did it in partnership with MBLEB.
You called it the Google Maps of biology. What's the scale we're talking about?
The scale is mind-boggling. Before this database, the total sum of human knowledge on protein structures was about 200,000.
Okay, 200,000.
The Alphafold database has over 200 million.
Million. Wow. So that's basically the entire known protein universe.
This is a 1,000-fold expansion of the map, basically overnight. And 3.5 million researchers are already using it.
This is the democratization part, then.
This is absolutely the democratization part. Imagine you're a scientist in a country without a multi-million-dollar X-ray crystallography lab.
You're locked out.
You were. But now all you need is an internet connection. People are using this for everything, from malaria parasites to enzymes that can eat plastic. And the data is free for everyone. Academic, commercial, doesn't matter.
I read that Pushmeet Kohli at DeepMind calls this a root node problem. You solve this one fundamental thing,
And you unlock entire new branches on the tree of knowledge. It's a powerful idea. But
I was waiting for the but. Here comes the conscience.
we still have to pump the brakes a little because while the map is huge, it's not always 100% accurate. And that new diffusion tech, the thing that makes it so powerful, it also introduces a very specific risk: hallucination.
I thought hallucinations were just for chatbots, making up facts.
It's a similar concept. A diffusion model is generative. It is built to create order out of chaos. But some parts of proteins in nature are genuinely chaotic. They're floppy, they're unstructured.
For ease of wet spaghetti.
Exactly. But the AI wants to create a neat, tidy structure. So it might denoise that floppy noodle into a beautiful, perfect-looking helix.
That doesn't actually exist in the real world.
And if you're a startup designing a drug to stick to that phantom helix, you're going to waste a lot of time and money.
So how do you know what to trust? Is there like a built-in lie detector?
There are confidence scores. And if you're going to use this data, you absolutely have to understand them. The big one is called PLDDT.
PLDDT. Got it.
It's a score from 0 to 100 for every single part of the protein. And the rule of thumb is if you see a region with a score below 70, you get skeptical.
What can below 50?
Below 50, the model is basically screaming at you, I am guessing here, or this part is probably disordered. Don't trust it.
Okay, so that's for one protein. What about when you have multiple things interacting?
There's another score for that, PAE, predicted aligned error. It tells you how confident the model is about the relative position of two different parts. You have to check that score too.
And I also read it sometimes gets basic chemistry wrong. Something about chirality.
Chirality is just handedness. Your left hand and your right hand are mirror images, but they're not identical.
Right.
Molecules are the same. Sometimes, because it's a generative model, AF3 will spit out the left-handed version of a molecule when nature only uses the right-handed one. It looks right on the screen, but it's chemically impossible.
Okay, so let's zoom out to the debate here. I hear all the caveats, I do. But from the builder's perspective, the business case is just screamingly obvious. This compresses R&D cycles like nothing else.
It does. But speed isn't everything.
But for a startup, it's almost everything. If I can skip years of trial and error in a lab and get straight to a high probability target, I'm saving millions. I'm getting to that go or no-go decision so much faster.
And I get that argument. Using it as a hypothesis generator is the correct way to think about it.
Right.
My worry is over-reliance. It's human nature. The computer gives you this beautiful high-res 3D model, and it feels like truth. It's very hard to force yourself to remember it might be a beautiful lie.
But no one is saying skip the lab entirely. The point is to narrow the haystack. You still have to find the needle, but now you're searching in a tiny pile instead of a giant one.
That's the ideal scenario, yes.
Yeah.
But we also have to remember this is a black box problem. We know that it works most of the time. We don't really know why the neural network made the specific choices it did.
True, but I'd rather have a map that's 90% right than be stumbling around in the dark. This tech is being used to design enzymes that eat plastic. This is bigger than just pharma. It frees up human scientists to ask the big what-if questions.
I agree with the potential, 100%. We just have to be disciplined and remember that the lab is still the ultimate validator. We have a map of the car parts, but we don't fully understand the traffic patterns inside a living cell yet.
Okay, fair point. So let's make this super actionable. For our listeners, founders, students, creators, let's give them a checklist.
Let's do it.
All right, first up, For students and researchers, what's rule number one?
Learn. 2.
Er,
Read. The PLDDT score.
hmm.
Seriously,
Oh.
do not build your thesis on a region that scores a 65. Treat low scores with extreme caution.
Okay, for the founders out there.
The database of single proteins is your gold mine. It's under a CC BY 4.0 license. You can build commercial products on it. But the new Alphafold 3 server has usage limits and terms you need to read carefully, especially if you're working with proprietary drug compounds.
Good tip. And for the creators, journalists, anyone covering this.
Please just stop saying AI solved biology. Say AI predicts structure. And the most important question to ask is always, has this been validated in a wet lab? If the answer is no, it's just a really interesting hypothesis.
Last one. For everyone.
Check the basic chemistry, remember the handedness problem. Don't assume the model got all the atomic details perfect just because it came from a Nobel-winning project.
So what we're looking at is, it's a future where the barrier to entry for biological discovery has just crashed through the floor. We have the map.
We do. But we still need explorers to go out and confirm that the map is right.
It's an incredible story. From a 50-year problem to a 10-minute starting point.
It is. A root node solved. Now we just have to carefully cultivate the branches.
If you remember one thing from this deep dive, remember this. Alphafold hasn't replaced experiments. It has given us a high-speed map to tell us exactly where to start digging. Thanks for listening.